Cut.
All right. This production was recorded at the podcast studios in the Media Arts Centre at Murdoch University. The views and opinions expressed in this podcast are solely those of the hosts and guests and do not necessarily reflect the official policy or position of Murdoch University. Any content provided by the hosts and guests are their personal opinions and are not intended to represent the views or opinions of the university, its administration, faculty, or staff. Now we have that out of the way, let's talk science. Paramount importance. Mussel Musterer.
That is me.
Do you have like a theme song to your...
No, but I would like one if you know any musicians.
I do know plenty of musicians, actually.
Okay.
Yeah, I think you should definitely have one. Today's guest, I've got Dr. Alan Cottingham, someone that I've actually worked with for a long time. Thanks for joining me.
No problem at all. My pleasure.
Did you know the very first project I ever did was with you?
Was it?
Yeah, in Toby's Inlet. And I spent...
That's right. Yeah.
Yeah.
That was crazy. That was the smallest boat I've ever worked on. And there was 4 of us on board.
It was pretty
3
funny.
units.
We had the water like coming over the boat if we all were like too close to sunset. I also spent that very first night. I spent in hospital that whole night and then had to come down.
Wow, I think you mentioned that. Yeah.
Yeah.
Yeah. We're lucky the river's only knee deep. So
it's knee deep of water and then about probably 2 meters of sludge. This is Toby's Inlet down in Dunsborough. But there was some good memories. I think that is what sparked my interest in estuaries anyway.
That's crazy. You need therapy.
We're not going to speak about estuaries today or fish. We're going to speak about mussels. So you are Dr. Mussel, mussel musterer.
Mussel man, something like that.
Yep. Physically, definitely. If you're listening to this, Alan, I've always described you a bit like a lumberjack. You're a big, tall, very muscly. But you're a very smart man and you've invented some cool things, which we're going to talk about today. But we're going to start off very broad. What is a mussel and how many species are there?
Okay, so that is a difficult question. You'd think someone that studies mussels would be able to answer what is a mussel quite easily, but as mussel is just really an English term that's used to describe a whole range of bivalves. Bivalves being a type of mollusk that have 2 shells attached by a hinge that sort of open and close depending on what the behaviour is doing at the time. And there are many different species of what we call mussels. So there's the true mussels, so they're the mytilids. There's about 400 species of mytilids. So these are the true mussels. Most of those are marine species. There's a couple in estuary. I think there's one that enters fresh water or lives in fresh water. And they have what we call beards, byssal threads that attach to hard substrates. We also have what's often referred to a mussel as river mussels. These are actually clams. They're very different to the mytilids because they don't attach to anything and they can move around with a foot and they can burrow into the sediment. I guess that helps them survive periods of drought. So
those clams that we see in the ocean, like the typical
giant
ones
clams?
Are they technically mussels?
No, so they're clams.
Oh, okay.
So the river mussels are actually clams, but we just call them river mussels because it's colloquial, easier to describe them as mussels.
Right. Okay.
Yeah.
Right. I actually did not know that. So Carter's mussel is actually a clam?
Yeah. So the Carter's freshwater mussel. So that's a mussel that's endemic to southwestern Australia. It's called a unionid, which are often referred to as river mussels. River mussels, there's about 900 species worldwide because they live in rivers, and rivers are one of the most impacted aquatic habitats on Earth. Around 45% of those species are red listed, so that means they're on state, federal, international conservation lists, such as the International Union for Conservation of Nature's red lists, either listed as threatened, vulnerable, near extinction, or extinct.
Right. Yeah. Well, same as I guess with estuaries as well. They're quite very rivers and freshwater places are heavily impacted, as you said.
Yeah.
Why are mussels important?
So they have a huge range of ecosystem services, is what we call them. So uh, one of the main ones in eutrophic environments, so environments that are got lots of nutrients, is filter feeding. So they'll remove lots of the cestin, so that's made up of phytoplankton and inorganics such as silt. They'll remove that from the water column, and then that energy then gets redirected to the benthos. So they're taking energy from the water column, they turn it into either a feces, which means they've digested it and it's come out the other end, or they've sort of bolted up in mucus and rejected it before they've even eaten it. And we call that pseudofeces. And because that pseudofeces, like the feces, has been balled up, it's much heavier than it was when it was floating around in the water column. So that also goes to the benthos, and that has huge benefits to macroinvertebrate communities in the sediment.
Right. So it's water purifying plus also providing food for other invertebrates.
Yep. And on top of that, they also get eaten by occasionally fish and mammals.
Yeah, right. Mammals. So I'm not surprised. I thought you were going to say birds, which I assume birds eat them as well.
Birds, intertidal birds will pick up bivalves definitely to eat. In the rivers, so the river mussels, one of the big impacts are like wild pigs that have been introduced.
They eat them.
Yeah.
Yeah, right. Not surprised. I've never made that connection.
Yeah, and a native water rat, although that as well
is a good thing.
It's not in as great as numbers as what it should be, so they're not having a huge impact.
Right. I've had some pretty funny experiences with water rats. They're for example, to explain what a water rat is literally kind of like a rat, but like 3 times the size. Like they're massive. We had one just chewing down on a mullet in the net one day and came on board. And my mate, who's rarely done any fishing or outdoor experience at the time, he just lost his mind. He jumped on the nose of the boat going like, what the fuck is that? What is that? And it was like, oh my God, it's a rat. And this ricali or water rat just did not care about the mullet at all. Did not care about us at all. Going back to the Carter's mussel, that is huge. That's a massive mussel. So like the different sizes of these mussels as well. Like you've got little ones, but the Carter's mussel is like almost the size of a fist. Yeah,
so the Carter's, the freshwater mussel, which is a clam, obviously, grows quite large. It's also really long-lived. Like it will live for over 50 years.
Right. What in comparison to other mussels?
So your mytillids, so the marine ones, probably about 4 years is a good lifespan for a mytillid. For the ones you get, for example, if you ordered chili mussels at a restaurant, they're Mediterranean mussels. They'd be about 1.5 years old when they're that big. It would take a Carter's freshwater mussel about 30 years to get to that same size.
Whoa.
Yeah. And
in terms of like, say, so like fish to reach sexual maturity, they have to reach a certain size. Is this sort of the same with?
So the slow to reach maturity, so it takes about 5 years.
Right. Okay.
Yeah. And one of the big problems we're finding in some of our rivers is that we find the Carter's mussels, but they're all large. They're all like 30 to 50 years old, and there's been hardly any recruitment for the last 30 to 50 years. So in another 20 years, for example, that's a big problem, right? Because the adults all die off and there's nothing to replace them.
Why? Why is that?
So they have a huge range of problems. So the Carter's freshwater mussels listed as vulnerable, and that's because of secondary salinisation. So as we've removed all the vegetation from the catchment, the salt table's risen. That gets washed into the rivers. Some of the rivers, like the Swan River, the Blackwood River, the Murray River, they don't have Carters because it's too salty.
But they used to.
Yeah, they used to. Yeah. And these are big river systems as well. The biggest river systems. And the reason why they don't have it is because they're big, because they often go into the scarp where the secondary salinisation is a bigger problem.
Yeah.
Whereas a lot of our other
So the
rivers.
scarp's like the wheat belt.
Yeah, the wheat belt. Yeah. A lot of our other rivers, such as like the Serpentine, the Harvey, they've only got relatively small rivers. They've got small catchments, and some of those catchments have still got their native vegetation. But of course, what happens when catchments have native vegetation is they put dams in, because that's what we use for drinking water. Whereas in the Murray River, you won't find dams because the water's too salty to drink.
So with the dams, that stops the flow going in, which then lets the salt intrusion come up the river?
So not so much, because we've got a pretty low tidal range, and the rivers are quite often higher than the ocean.
Okay, so then what's the impact in these? They're obviously slowly getting degraded.
Yeah. So
is this just the build-up of nutrients?
Yeah, so obviously the salty ones, no mussels, too salty. The other ones, you're stopping river flow, you're putting in barriers. One of the really cool things about river mussels, and this is river mussels worldwide, so there's 900 species. The Carters is one, I think, there's a subspecies of Carters in southwestern Australia, but let's just say it's the only species in southwestern Australia. Like other river mussels, fertilisation for reproduction. So the male releases the sperm, and the female takes it in during filter feeding. It's then everything sort of happens internally. They then release.
So the sperm is free swimming in the water?
Yeah. And
it just happens to find the female? Like, how does the female catch? Are they got to be close together, or?
Probably not. So they're quite fairly highly fecund. So they release quite a lot of it. Yeah. And it's very similar to broadcast spawning. So most mussels, like marine mussels, the male will release the sperm, the females will release the eggs, and then they rely on those meeting in the water column. So it's very similar to that, except the female doesn't release the eggs. Fertilization's internal. They then release the eggs, the larvae called Glaucidia, in these little mucus strands. And the mucus strands just pretty much float attached to the mussel, and then it takes a fish to swim through that mucus strand. And the little larvae, the Glaucidia, they look like a little clam, but they've got these massive teeth. And what they do is they latch onto the fish, because that Glaucidia stage is a parasite. So they need a fish host to swim through, and they'll grab hold of that. So the Carter's freshwater mussel, the Glaucidia is external. It actually looks like a little pimple. You can see them quite often on freshwater fish. That freshwater fish will then, usually migrate, usually upstream. Of course, with dams as barriers, so they don't always get to where they need to go. And then after a month or so, the little Glaucidia will drop off. It will then live in the sediment for months. So you need really good chemistry in the sediment for these to survive.
How big are they at this stage?
So they're still very tiny, like
less than a millimetre.
Yeah, maybe a millimetre.
Oh, right. Wow. Yeah. Okay.
And then they'll live in there for months, and then they'll slowly come out to the surface, where they'll start their juvenile phase. And then about 5 years later, they'll reach maturity, and the process will start again.
They move down the river from after that, or?
So we're not sure exactly.
Okay. That is wild.
Yeah. I mean, one of the crazy things is that so these river mussels are really important. In North America, I think 60% of them are red listed of the 300 species, but we don't know that much about them in general. For example, we know, like in drought, they've got really two options. They can move or they can burrow, but we don't really know what that decision is based on. So, and that's the difference between life and death. If you go to move, but if you move to the wrong spot, or quite often, there's snags and stuff, so there's barriers that stops you from moving, you can end up being out of the water when the river dries out, in which case it's pretty much over. If you burrow near the riverbank, you could be having to stay there for like 4 or 5 months until the river comes back. Whereas if you burrow right in the middle, you might be there only for a month or so before the river, the water comes back.
Right, and that obviously quite relies on that the water quality in the pools that they move to have to be good as well, because that's usually not the case when the pools dry up. How long can they survive with, say, deoxygenated water? That's usually what happens to these pools, right? They become
Yeah, so I'd say deoxygenated water would be very similar to not having water at all.
yeah.
So that would be about 2 months for carters.
Mm.
There's mussels, freshwater mussels in Scotland that can live for over 2 years, just buried in the sediment because it's much colder.
Right. So they don't dry out.
Yeah, well, if they dry out, then yeah, they'll die.
Yeah.
But because they close up
Yeah.
and they retain that moisture inside their shell, if they lose that, then yeah, that'll be it.
What's the like limiting factor for them? Is it like they need water to breathe, like oxygen, drying out, or like what's?
Yeah, so they're able to close up shop and just sort of hibernate or whatever you want to call it for months.
Right.
And yeah, and once the water comes back, they'll be opening up pretty quick because there'll still be, if they're feeding before they close, and they'll still be excreting probably inside their shell, which wouldn't be very nice. So as soon as that water comes back, they'll be opening up and trying to get oxygen and food.
Right.
Yeah.
What's the salinity preferences for like these mussels? Because you mentioned the Carter's mussel it's getting too salty for them, so they can't breed anymore. What is this? I remember when I went up to Moore River, I messaged you going, "Why am I not seeing any pygmy mussels?"
Yeah,
and you said there's certain salinities, and then that made sense. So what are these salinity ranges?
Yeah, so salinity, as you know, is probably the driving factor for the ecology of most fauna in estuaries, rivers, and the ocean. So the Carter's freshwater mussel, if salinities get above 3 parts per thousand, so that's, seawater is about 35 parts per thousand, so 10% of seawater, you get really high mortality.
Right. And you said the pygmy mussels, like, because they can survive, adults can survive a larger range, but their juveniles can't, and they can't breed. So what is those ranges?
Yeah. So the pygmy mussels, they're one of the true mussels, the mytilids. They're an estuarine species. They've evolved to breed when salinities range between, I think it's about 7 and 32 or something like that. So it's a wide range of salinities they can actually breed in and successfully recruit. But quite often in estuaries, especially ones that become close to the ocean, salinities are well above seawater levels for most of the year. So, and because the pygmy mussels are short-lived, like most estuarine fish species, like it's a 1 to 2 year life cycle. If you have, a few years of high salinities, and that'll pretty much wipe out the whole population. So while the early stages have that window of around 7 to 32 salinity, the adults, that they're quite happy to go up to about 60.
Right. Yeah.
Crazy.
Well, Moore River was the opposite case. That was too fresh.
Yeah, so too fresh. So you're not getting that the lower end of the salinities for spawning.
Right. So you've compared the filtering capacities between different mussels. So how much water can they filter? Like why? Because obviously it sounds great when you say, they're pulling the organic nutrients out of the water column and all that. Like how effective are they?
So they're incredibly effective at water filtration. So the little black pygmy mussels, now these guys are probably 20 millimeters, probably 50 millimeters, something like that. They're quite small. And a mussel that size can filter about 100 milliliters of water an hour, which doesn't sound much, but you can get densities up to 100,000 mussels per square meter. So when you times 100,000 by 100 mils, you get 10,000 litres per hour from 1 square meter of mussel bed.
This is going to be a hard one. Do you know the volume of a Olympic swimming pool?
No.
Me either. I'm just trying to think of a way and put it in comparison. Like how like that's because that sounds like a lot of water, but I don't know the volumetric like comparison to say how much water is in an estuary. Like is it clean?
Yeah, so I think for example, the pygmy mussel, because we were doing habitat enhancement, so we're putting out koi matting, and the mytilid mussels, the larvae pretty much need hard structure to attach to recruit. So we put out, and many of our rivers have had lots of snags removed for, water flow and preventing boat collisions. And there's a lot of initiatives now to actually start putting all that sort of materials back in. But the pigmy mussel larvae will attach to the hard structure, and one of the hard structures they seem to love is like the coir matting, which is like a coconut fibre husk matting. And we did some trials with that, and I think we found if you put out 1 kilometre of that, it's going to filter the whole river, within quite a short period. I can't remember what it was, but it was quite impressive.
That is very impressive.
Yeah.
I've helped you out in a few of these, when you've gone out in the field, and to be honest, I probably was more just playing around with the mussels than actually thinking a lot about the science behind it. That's incredible. Yeah. Now I want to expand on that. You're in a lot of these restoration projects. We'll talk about these ones specifically what you just mentioned about the restoration. So this is all in the Swan River and the Murray?
Mainly the Swan River.
Mainly the Swan River. So what has been happening?
So over the last, say, 8 years, I've probably been involved in 3 main shellfish reef restoration projects. The first was one of my honours students, Charlie Maus. We didn't have much of a budget, so we got jarrah stakes, and it's called a bow shot style. It's something that the French used to actually cultivate mussels for food. Is we put the stakes covered in mussels into the substrate and about 3 meters of water in Melville water. And Charlie monitored the mussels, looked at the body condition, the growth, and looked at all the other the fish that used those habitats, and also all the other macroinvertebrates that became part of that community.
I came out with Charlie a few times, and he was having to dive in the Swan River all through the year, and diving in a river is always quite not the nicest conditions.
No.
But one of the problems he had was a lot of the mussels were dying.
Yes.
So why is that such a big problem to keep mussels alive? There's also the other shellfish reefs that you're building, being a part of, I should say.
Yes.
Why is keeping them alive been so difficult?
Well, mussels, like most animals, have a set of variables that they like to live in. Unfortunately, with some of the projects in the Swan, is that because of all the other recreational use zones in the Swan, doing stuff in the shallow waters, which is where mussels actually prefer to be, is very difficult. So all of our restoration activities have been in the deeper waters. Mussels love flow. There's not much flow in the deeper waters, and the reason why they probably love flow is because they filter so much cestine out of the water that they're basically creating this continuous stream of poo. So if that just, if you're a mussel bed and you're just pooping continuously, then you're just actually smothering yourself.
Don't shit where you eat.
Yeah, exactly. So there's that. There's also, unfortunately, the mytilid mussels that we use for the shellfish reef restorations on the hard substrate, the marine mussel, is pretty much at its most northern temperature range in the Swan. So when you get a bit of a hot summer,
like this year,
like this year, so when temperatures get above 24 degrees, water temperatures, they start to struggle. 26 degrees, they really drop off, and I think we're getting temperatures up to 28 degrees or something.
During the Fish Index project, we've been measuring 30.
Yeah,
30 degrees for, and that was also last year as well for periods, which was significantly warmer than we've ever had.
Yeah.
Because that's also impacting the fish that's in there, we're finding now as well. So obviously it's also the invertebrate communities as well.
Yeah. So, one of the things we are looking at for the Swan is how to future-proof the reef restoration initiatives by looking at what other species are there that could potentially be used for restoration. Unfortunately, quite often some of those species aren't native to the Swan River. Some are native to Western Australia. Some are just native to Australia. But of course, all these borders are just borders that we've put in. They're not ecological borders for species. So Eventually, those species will probably get here.
When I was up north on that last project being on, there was massive mussel beds in front of the creeks up in the Pilbara.
Yeah, right.
They looked like the blue mussel.
Yeah, they all look very, very similar. So that would be a different species.
Right. But would there be a problem with, like, say, not like translocating them, like, or using them?
There is. As humans, we've introduced a lot of species, and often to great economical costs. So there needs to be a lot of debate around introducing species that aren't native before anything like that goes ahead. And I doubt it ever actually would.
Yeah,
I think it would be more of just let it happen organically.
Yeah. Okay.
Yeah.
Right.
And we do see during all our surveys, we see lots of different species. Some are tropical. We seem to find that in the summer, they'll be there. In the winter, they'll die because it's too cold. And then you get some of the temperate species that you'll see during winter, and then in summer, they'll die off. So
no happy medium.
No. So Perth's unfortunately at that interface of tropical and temperate.
Yeah. Well, this year, as you've seen, a lot of the videos we've been putting up, a lot of tropical species around coming down, and a lot of people, they're going to survive here. And it's like, probably not.
Probably not. Maybe another 50 years and winter will be a little bit warmer. And we'll be seeing more tropical species survive.
Yeah, but we're also going to see a lot of temperate species not survive.
This is true. Yeah. And look, the reason for that is marine, so ectotherms. So these are animals that can't regulate their body temperature, unlike endotherms, humans, birds, mammals. So we have a huge range of temperatures that we perform optimally at. Whereas if you're an ectotherm, it's usually a very single temperature. And if you go beyond that or below that, then your potential for growth and everything decreases. So these are the species that are most impacted by climate change and marine heat waves and, these sorts of events.
Right. Yeah. Crazy. So what are the main predators of these mussels? Because that was also one of the other reasons why these mussel reefs weren't surviving. Because I remember when you put out mussels at a few centimeters or a centimeter or a few millimeters size, and there always seemed to be a predator that loved each size, it was a sad but funny story. If in a way, because you'd put we'll put them out this size, and then I'll let you say it, but like seemed of like, oh shit, these ones wiped them out, and then the next one we'll put bigger. They didn't eat it, but then something else did. What?
Yeah, I mean, we had problems. I think the first initiative was that we had some spat rope that we had hanging in Coburn Sand. So Coburn Sand, up to a couple of years ago, and this was put out in the 90s, was a aquaculture farm for mussels. And that process is the mussel farmers hang out spat collection rope. The spat is the mussel larvae. The mussels are highly fecund, so they do that broadcast spawning thing. The larvae then look for something to settle on, which is the spat rope, which is almost similar to the coconut husk fibers that we're using for the pigmy mussels. And they'll settle on that. And quite, if they're for farming, then they're left there. But we were taking those when the mussels were probably like a couple of millimeters, and we're just stringing those over some of the pilot reefs, the trial reefs that went in before the big, the larger scale reefs. And we put those out, and I'm pretty sure within like 24 hours, they were all gone by blowfish. Yeah. So then the
fisherman's worst nightmare too.
Yeah, that's right. So to get over that, we did a couple of quick little trials, and we found that the mussels get to about 10 mils. The blowfish seem to like pick them up, spit them out. They can't sort of break through them. And then after they do that a couple of times, we just left them alone. So we did some trials just in baskets with a camera, but the blue swimmer crab seemed to think that was a pretty good size for them. So they would come in and pretty much clean out those. And then I think we went to larger mussel again, and that one seemed to be quite quite a good size for stingrays. And there was a lot of eagle rays, and there's a lot of eagle rays in the Swan, as you'd know.
Yeah. Which as you would know as well, back in the earlier days, like 70s and all that, I think in 10 years of sampling the Swan during the 70s, they caught one eagle ray.
Yeah.
And then as the decades and the estuaries become more marinized, which is like, become a bit more like a marine embayment, now we're catching hundreds and hundreds of eagle rays, which is like insane. But yeah, they're when cause there's still a lot of blue mussels in the Swan, but they're like, on the moorings and things like that.
Yeah, moorings, usually shallow water, where there's good flow. Even in Coburn Sand, so the mussel farm, so they were having their grow out ropes to going down to about 8 meters, I think, without too many problems. One thing they did find though, if mussels come off the lines and fall to the ground, they get eaten by crabs and stingrays. On the ropes, they were fine until the snapper. The snapper would come in around November after spawning, and they would clean out, like tons of mussels. So in the end, the farm stopped producing mussels. One of those reasons was the snapper eating all the mussels. The other reason is you might have heard from Department of Health, don't eat recreationally caught mussels. This is because, for example, the mussels at the mussel farm in Coburn Sound, they were spending $250,000 a year on testing the water for different types of algae, testing the water for other contaminants, and also testing the mussel tissue to make sure there's no bioaccumulation of heavy metals so that they're basically safe for consumption.
And that's why in the Swan, do not eat the mussels.
Do not eat the mussels in the Swan, especially around yacht clubs, because that's where tributyl tin for biofouling.
Also, all the leakages of petrol and fuel. So they're filter feeders, so they're taking all the nutrients out of the water.
Yes.
And then, as you would know too, estuaries are sink for nutrients, literally. They're the lowest depression in the land, and everything that falls in the catchment, which is for swans, was it 2 times the size of Tasmania or 3 times the size of Tasmania?
The catchment, it's quite big. I think it's, something like half the size of Victoria.
Yeah. So everything that falls in that area eventually washes up into the Swan. So, and then the mussels filter that out. So do not. Well, that's why there's a lot of signs of do not eat the mussels.
Yes.
Going back to that project, is it still ongoing? Still finding ways to?
So the project officially ends in June this year. So the Nature Conservancy have finished. They've put out the reef. They've done all the monitoring that they said that would happen. The results aren't as good as what they'd hoped, which is, it's quite normal for restoration projects. It's not really a failure. The reefs themselves are like amazing. So even though the numbers of the Mediterranean mussels, which was the focus of the project, are quite low, was they're seeing like lots of abundance of scallops, oysters, even Angazi, the flat oyster. So the flat oyster, it's iconic in that before European settlers, or when European settlers came to the Swan over 200 years ago, there was all these dead shells of Angazi flat oyster, so the flat oyster reefs in the Swan. So they dug them all up. That's what a lot of the buildings and roads were made out of in Perth back then. And no one's really seen live Angazi flat oysters in the Swan, but we're seeing them. We put structure back in, and you're getting some recruitment of the flat oyster.
No way.
Yeah.
So that's awesome. That's a huge positive response.
Yeah.
Maybe the mussels haven't quite worked out, but all these other things, because they're all filter feeders too.
Yeah, well, that's right. And I mean, not only are those bivalves, but sponges, sea squirts, even algae growth is, it's sequestering nutrients and it provides habitat and food. And when you look down there, like you can't see much further than half a metre just because of the fish. There's just incredible numbers of fish.
All right. Awesome. And those fish are Well, I remember putting ROVs on it. We're mainly gobble guts and
There's lots and lots of gobble guts.
Yeah,
They love that habitat.
Yeah, they do. What's the filtering capacity of these, like of scallops and the oysters and the sea squirts? Have you measured them as well? Or?
So sea squirts would be even higher than mussels.
Right.
Those guys, they're pretty crazy, but I don't think anyone's really done that before. And oysters have been done. So a large oyster, they reckon about 9 litres per hour, which is huge. But that is a big oyster. So, mussels are probably a mussel reef, because you get higher densities of mussels, and because of their filtering capacity being only slightly less than oysters, the mussel reef would have a greater filtration capacity than an oyster reef. The other thing about oyster reefs, oyster reef restoration projects worldwide have been, what most people have focused on when it's shellfish reef restoration. But of course, trying to grow oyster reefs to the extent they were hundreds of years ago would take hundreds of years. Whereas mussel reefs, you can pretty much, in a couple of years, you could have, fully functioning mussel reef.
Right. Okay.
But the problem with that is if you have a perturbation, such as a low oxygen event or something like that,
it could all get wiped out.
You'd get wiped out. You could go down there, you would not even know the mussel reef was ever there. In terms of the mussels, the shells, they dissolve or get washed away. Whereas at least the oyster reef, you still have an oyster reef, there just wouldn't be living oysters on there.
Yeah, okay. Why do they dissolve so quick? Because they're so thin?
Yeah, they're quite thin.
Oh, okay. Yeah, interesting. So you're a scientist, but you're also a bit of an inventor.
Well, definitely not an inventor, is that. So,
I reckon you are.
I'd say innovator.
Innovator? Well, you invent things, you build things, and you've come up with a very cool project. So this is, and I am going to talk about spy valve now, which is, it's just a way that you're also monitoring the health of these, using mussels to monitor the health of estuaries and rivers and all that. So please elaborate.
Okay, so project spy valve. So it's a bit of a play on words, right? Bivalve, spy valve. So spy was sort of spying. Valve, obviously the mussel shell. So we're using mussels to spy on human activity. So the project uses sensors attached to the mussel shell. If the mussels are open, it means they're happy. It's how they feed, breathe, excrete. If they, and this is most of the time, 99% of the time, they're pretty much open doing this. This is what they do. They only sort of close if there's something in the water that they don't like. So we use that behavior. If all the mussels at a station, for example, close, we go, okay, there's a problem. And just to be clear, so this type of approach has been used for a long time in Europe, even since the 1990s. There's a really good YouTube video on Poland's water reservoirs. So in Poland, they're drinking water reservoirs protected by 8 clams. Of course, they've got all the other fancy equipment that you'd expect to ensure freshwater security. But as a last level of defense, they have a tank with 8 clams with sensors attached. And the reason they do this is because there's over 300,000 industrial chemicals on global markers. And you can't really measure, you could probably measure them all, but it'd be very difficult. And, these aren't just things you can put a probe in and go, the concentration of this is that, right? You need a PhD in chemistry to actually extract.
And it takes probably months to get the results back as well, right? Which is, if the flicks like that and the water is contaminated.
Yeah. Don't drink that 3 months ago.
Yeah. Exactly.
Yeah. And then, of course, we don't really know what happens when some of those contaminants actually mix with each other.
Yes.
And look, a really good example of this is in America, there was mass mortality of salmon. Right? Salmon are a really important species over there. And you're having these millions of salmon die every year, and they couldn't figure out why. And it wasn't that long ago that they realized that what was killing them was this, it's called 6-DDP quinone. And what it is 6-DDP is a preservative that they use in car tires. When you drive down the road, little bits of rubber come off your tyre. That preservative, I think, relatively harmless. But when it comes into contact with ozone, which is quite common in summer at ground level, it forms this compound that's highly toxic. So when it would rain, that would get flushed into the rivers and pretty much kill everything. But they weren't testing for it because they didn't even know it existed.
Is ozone natural?
Yes.
Right. And these would be the same car tyres, like
worldwide, yeah. So they've tested for this compound in Queensland, and they've of course found it. I think you'd find it pretty much everywhere you tested for it.
Right. Is it in dangerous levels anywhere else apart from there?
Well, no one's.
Why was there so? Why was it so high there?
I'm not sure. I guess a lot of people drive, a big population, a lot of roads.
Didn't you think it would be happening in, like, say, China and India, and probably their waters don't have as many fish in it, I guess, maybe.
Yeah. But there's also, quite often when there's a storm event after a long period of a dry spell here, we get fish kills, and we often go out to the black water or it's the low oxygen. But of course, what happens with fish kills is when a fish die, it actually sinks. It then starts to decompose, and during that decomposition, it's microbes inside the fish that are producing carbon dioxide or methane or a gas that actually makes the fish float to the surface. And it's not until then that a community member might see it and go, there's some dead fish floating here. And then, they have to bring the right people, of course, and then DWER, so Department of Water and Environmental Regulation, who are responsible for all the inland water bodies in southwest Australia. They'll go and investigate. But of course, by the time that's even reported, it could be a week too late. So that fish probably died a week ago. It could have died 20 kilometres upstream. So then trying to infer, okay, what killed this fish? They'll take water samples and measure. Of course, quite often, the oxygen's low here. Well, you've got a ton of dead, decaying fish, so it probably would be low. But what was it when the fish actually died? So what we're doing is we're using these mussels, so they're attached to the sensors. We've got them at field stations. And we
say fuel stations?
Field stations.
Oh, okay.
Yeah. We're using different technology to what they're using in Europe. The sensors in Europe, because they've been pretty much using the same approach, same technique, same sensors since the 90s. And these are sensors that you have one sensor that creates a sine wave, the other sensor reads that sine wave. And even when that sine wave's low frequency, like 1 hertz, which is one thingy per second, something like that.
Yeah.
They're taking like 1,000 readings per second to recreate that sine wave to actually go, okay, is the clamshell open or closed? So that's a lot of data.
Especially if millions. You've got multiple mussels as well.
Yeah. And there is a project that's called Molluscan Eye Project that's come out of Europe as well. They've got field stations throughout the world. So they use the same type of sensor. What theirs does is at the end of each day, the data, millions of data points are uploaded, sent to a research assistant, I think, and he's got to sort of download the data and analyze it, work out what's going on. So, and they have half a dozen professors working on that project. For the spy valve, it's been just myself. So, of course, I need everything to be automated.
And in your own time, a lot of it.
Yeah, in my weekends and after school.
Yeah.
So I need it to be fully automated. So I use a sensor that other people have used before for this type of study, and they're called Hall effect sensors. And what they do is they measure magnetic field. So on one shell of the mussel, I have the sensor. On the opposing shell, I have a little magnet. And the sensor, all it reads is whether or not what the magnetic field is. So the old school Hall effect sensors, if you think of it like a little cog, so when you turn up your stereo, you're increasing the voltage, makes it louder. So if you think of it as inside the little sensor, there's a little cog, and when it gets closer to the magnet, it turns. So it varies the amount of voltage. So the sensor is just reading the voltage.
Mm-hmm.
So depending on that voltage, we know whether the muscle's sort of open, fully closed, and, in between as well. Because sometimes we'll do little micro closures, which is they'll close sort of halfway and open again. So we're just learning about all these different behaviours. Of course, if all the muscles, so each of our field stations, we have 8 muscles. If all of those muscles close for 5 minutes, and that's what they use in pollen as well, excuse me, we'll go, okay, there's a pollution event.
And
It's
a immediate response.
Yeah.
Yeah. Compared to 3 months.
Yeah, compared to, yeah. So if all the muscles close, I'll get an email, and then I'll go to the next stage. So this project, the current project is funded by the Alcoa Foundation. It's called the Alcoa Foundation Cybernetic Water Quality Monitoring Program. And we've got 6 stations. They're all in the Pilharvey region. Each of those stations linked to a school group and some Indigenous rangers, with the idea that they've been receiving the training to go, okay, so we've got this early warning system, but we also need an early response system. So community groups, because they're right next to the estuary, can actually be the people that go down and go, yep, I can see that there's fish trying to gasp oxygen at the surface. There's a problem with the water, and then de-work and then get down there as the fish kill's happening and take all the samples they need to actually try work out exactly what the cause is.
Right. That's amazing. Is this like, I know you said there's other projects similar elsewhere in the world, but this is probably the first like this, right? We've automated.
Yeah, this is the first.
And anyone can see, because this is on a website online as well.
spyvalve.com.
So anyone can go online and see how the mussels.
Yeah, see the mussels at those 6 stations.
Have you ever thought about getting like people to like sponsor a mussel?
Yeah, I mean, that would be a great idea. I mean, I'm looking at, expanding, obviously.
Yeah.
Beyond the Pilharvey catchment.
Because we have multiple estuaries where there's been fish kills. There's been a few recent ones on the south coast that I've been a part of. Yes. I didn't cause them, sorry. But, been alerted about them. But yes, but you could essentially implement this anywhere.
Yeah.
How like expensive is this?
Well, I did some back of the envelope calculations, and it would cost this. So each station has about 8 mussels, say 8 mussels, but it would cost less than a cup of coffee per mussel per day.
Wow.
Coffee is expensive though, right? So might not be the best example.
So if I wanted to say I'm a philanthropist.
Philanthropist,
yeah, that one.
I like philanthropist.
And I was like, I live on a river, or I've got a certain interest in a specific river. I want to sponsor a station here. What does it cost to set up?
So it'd probably be a couple of $1,000 to set up. That's for the equipment. And it's also depending on, so because the data is pretty much live streamed. So the most demanding part of that is the dongle, so the internet connection. So because of that, you need solar panel battery. But if you've got your own jetty that's got a power supply, then you're laughing, because that's more than half the cost.
Right. That's very, very feasible.
Yes.
I can even imagine farmers, like, the farms that have grown up on family, we have rivers going through there. I don't think they're all fenced off, so the cows can't go and drink out of them.
That's a good thing.
Yeah. Yeah. But I can imagine there's definitely I definitely see a lot of cows on rivers. Usually they're all salty in the areas I'm in, but I can imagine there's just so many different avenues that people would find this interesting. And that's a very feasible thing. Like, I think hopefully there's at least one philanthropist. I'm going to stop.
One person with lots of cash.
would find that important to, if that could be set up. That's
So interesting you mentioned cows, because one of our stations, we put it out. We thought, okay, this looks like a good spot, and we were lucky enough to have some access to a private farm, and just downstream of the station is a river access point for cattle.
Mm-hmm.
And when cars stand in water, they actually poo 5 times more frequently than when standing on land.
What?
And that was a study done in Europe when, you have a daytime temperature, maybe 25 degrees and a water temperature of 10, whereas we've got a daytime temperature of 45 degrees and a water temperature maybe of like 20. So when they put their feet into that colder water, it just makes them go,
No way.
yeah.
I had no idea about that.
Yeah. So, and they love standing in the water.
I'm gonna tell my grandparents and uncle and cousins on the farm that's what they do.
Yeah. So so this station, we're also partnering with Department of Water and Environmental Regulation. So they've put out normal, traditional water quality monitoring equipment. So like dissolved oxygen, temperature, salinity. And one thing we're seeing is pretty much for the whole summer, the mussels stayed closed and DO was pretty much donuts, 0. Which is strange, right? Because coming from, my background's in estuaries as well, and quite often if you have high nutrient load, you'll have supersaturated oxygen during the day because you've got lots of nutrients for phytoplankton, and then at night it'll go down to 0 and then it'll come back up. And you've got all this crazy stuff happening with your pH and everything changing as well because of the production of carbonic acid and, lots of different things going on there. But for some reason, this water, it must have had such high levels of nutrients and stuff happening that not even phytoplankton were going to take advantage of it.
Right. So it exceeded their limits.
We haven't tested, but, to come across water that's got lots of nutrients and 0 oxygen, even on the sunniest of days, is very, very strange.
I can imagine the mussels didn't survive there.
They actually did.
Oh.
Yeah. Yeah.
How?
So, well, because of the Carter's freshwater mussels, they can shut shop and live for several months without anything.
Right. Wow.
Yeah. So as long as they're moist and cool. The study that was done by Al Limbri found it was at least 65 days. The study didn't go beyond that, so it's going to be longer than 65 days. But of course, when a river dries out, if you're not in the water, if you're not in the sediment that's moist, and you've got to stay there for 6 months, you're probably going to die. And one of the problems is, of course, as you know, declining rainfall, water extraction, damming. So a lot of our rivers, especially where you find the Carters, are actually, they've become these disconnected pools. And a lot of those disconnected pools, they're extensions of the groundwater table, but evaporation is so high, they're actually quite often, the groundwater table is actually higher than the level in the pools, which is bizarre. It just takes a little while for the water to get back into the pools.
Yeah. I remember working up north with Matt Hipsey, and he was telling me how fast or slow, depending on the sediment, which a lot of estuaries and areas like that are made out of clay, which is the least porous.
Yeah.
Then it was I wish I could remember actually what he said.
Oh, yeah, it would take a long time.
Yeah, it moves really slow.
Yeah.
Right. Can you restock like the Carter's mussel? Like, can you just get like that phase, that early, really vulnerable phase? Can you just bypass that, or that part where they become a parasite's impossible to? Yeah.
So we're looking at a project to restock Carters, or at least do a pilot study. So pilot studies where there's a proof of concept to say it can be done. And in theory, it should be quite easy because all you need is in winters is when they spawn. The larvae, they're ready fertilized. They're in mucus strands. They're actually quite easy to collect. And then you can just put those in a bucket, put some fish in there. Native fish are probably better hosts than, for example, gambusi. Let them swim around. The fish will come out covered in glaucidia. And then you just have to keep the water quality good for the fish and the glaucidia.
It's a bit bigger than a bucket.
Well, The bucket stage is initial for the Glaucidia attachment, and then you'd put it into like a larger aquarium.
Mm-hmm.
And we're looking at trying to do that with John Tonkin College. They've got a really good aquaculture program there, and they've got lots of tanks, so we can actually do proper trials with different fish, different sediments to see which works best, because we don't really have that knowledge yet.
Okay. That's exciting that it's happening though.
Yes.
That, especially if they haven't bred for 20, 30 years.
Yeah.
That's
crazy.
wild. So awesome that it's happening now before it's too late. I think my next questions probably could be a whole podcast on itself, which I won't let it go that long. But both you and I, we've done a lot of field work, and on the riverbanks and estuaries, you've come across some very strange things.
Mm-hmm.
What are some of the stories? Did you have any you want to share that you see? Because
it's only some I probably can share.
Yeah.
So look, peri-urban environments, so they're like the areas between rural and urban areas, and they seem to be, some of the worst places I've ever been, because they're often graveyards for stolen cars, burnt out, pig hunting camps, which can be pretty scary to come across, because you know what pig hunters are like, and you know that what their dogs are like. Could end up in tragedy. But abandoned mobile meth labs, I think, are probably one of my favourites. It's like you come across them, you think maybe someone's just camping here, and you sort of look around, you think, I don't remember last time I was in a camping store seeing these bottles of corrosive liquids.
Yeah, right. Yeah, I actually don't think personally I've come across that. Maybe just without knowing. I'm usually on the water on a boat.
Yeah,
not so much.
It's not really until you actually have to, hiking through sort of woodlands looking for old rivers, for example. A lot of our rivers have been diverted, but some of the rivers are still extensions of the groundwater table. So when we're doing our mussel surveys, we're also looking in the old parts of rivers just to see if we can come across mussels there, as well as the new diversions.
Yeah. I know that you've got a lot more other stories as well. Did I ever tell you the story of when Charlie and I were up at Guildford with the during COVID, and the lady with the bommy knockers and the whippersnipper arms?
The topless lady?
Yeah, the topless lady.
Yes. But tell me again.
Yeah, I think everyone, whoever's got to this far of the podcast would probably want to hear it too.
Might be the highlight.
I know. The information you've told me, it's blown me away. I've been working so close to you for so many years that I can't believe I didn't know all these things. But yeah, so this topless lady. So this is going back to 2020 in COVID, during peak COVID time when you're allowed 1.5 meters away apart. Charlie and I were doing the fish index project on the Swan River, which looks at the health of the estuary by looking at the fish communities. So to do that, we have to go fishing with nets. Anyway, so we're up at Guildford, and Charlie and I pulled up in the boat, and there was this pretty big burly bloke, big silver beard hanging down, had a blue singlet on, a few tats on his arm, drinking a yellow Bundaberg. And then next to him was a lady, and she was one of the ladies that she looked about 60, but I think her biological age was about 40. So, a few missing front teeth kind of style. And all she was wearing was underpants.
Thank God.
And her arms were going side to side like a whippersnipper. And the other parts were going side to side as well, and they were quite long and stretchy. Anyway, I pulled up there, and the bloke's like, "Oh shit, I thought you were the cops." I'm like, "Nah, mate, you're all good." And then she yells out, she goes, "How you going?" "Oh, good, thanks. How you going?" It's like, "What are you doing?" And anyway, so I told her what we're doing. Like, "Oh yeah, we're fish index. We're looking at the health of some river, looking at the fish communities." Anyway, so Charlie and I went and did the seine net, and so the 21 meter net, and you pull up and then you pull it on the bank, and then there's all these little fish that are inside the bunt. Anyways, we started like pulling them out and throwing back the brim and the Perth herring, and then she's come over, still with the whippersnipper arms, and she's probably within about a metre of me now, standing there. And then she's like bent over and her arms turned into windmill arms and her big saggy boobs,
for lack of a better word.
yeah, they turned into like well, I called them bommy knockers, but and I was like dodging them, going side to side, and she's like, "What's that one? What's that one?" I'm like, "Oh, this is a broom. Oh, this is a bird herring." Charlie couldn't look at me in the eye. He looked up once and almost pissed himself laughing. He's down there just not even looking. I'm trying to speak to her, so moving my head side to side, trying to dodge. And she was there for about ages. It was so funny. I was trying not to laugh, and she was just, her arms did not stop. Her tits did not stop swinging. That was a pretty funny experience. I guess we obviously packed up and did our thing and moved on. But yeah, there's definitely see some funny things on the river.
Yes.
I do have one last question. This is probably going to be, these are the my ending question.
Yep.
What is your least favourite mussel?
Well, I guess it's probably the zebra mussels, the dracinids. So
These are introduced ones.
Yeah, so they're an Eastern European species, but they're introduced in the Great Lakes. So they haven't hit Australia yet, but they're a freshwater mussel, false mussel, so they're not a real mussel, but they identify as a real mussel if they're allowed to do that. But they actually cover, so our friends, the river mussels, so they actually biofoul the river mussels and stop them from burrowing. So they knock them off, basically, if there's a drought. They can't burrow to protect themselves.
Oh, gee, right.
Yeah. Bastard mussels.
Yeah. Fair enough. I don't like them either.
Yeah.
Are they in the estuaries?
No, just freshwater. But we don't get them here yet.
Right. So that you said they were introduced. Is that saying they're like actually brought over on purpose, or they just...
No, usually in ballast water. So that's,
okay.
Most mussels.
Which is a ship though. So how do they get into freshwater?
Yeah, I guess the ships must somehow navigate in there. I'm not sure.
Into a lake?
Yeah. I don't know. I actually Googled this. I was like, but it just says ballast water. I'm like, well, how does that happen? So there must be some way of doing it.
Right. Okay.
And all the pages say the same thing. So.
I'd say that if they've if they made it across by the ballast water in weed, ducks eat weed, or on their feet, weird, or right? Ducks or something.
If they've got a salinity tolerance of close to zero because they're fresh water, then that ship must have done a transatlantic voyage but come from a freshwater.
Yeah. Anyway.
Strange.
It is strange.
Yeah. But mussels in general are one of the most species that are introduced around the world, even been introduced on every continent, even Antarctica.
Oh, really?
Yeah. And that's because of that ability to, well, a lot of mussels love the hard structures. So they'll attach to ships, and we're moving hard structures around the world, so you can get them on the bottom of ships. They also have potentially a long larval stage of several months, so they can swim around looking for somewhere to settle for months. So if they end up in ballast water, they can come out at the other end and they're in a port, which is hard structures everywhere. So they're pretty much good to go.
Because these are mussels, they also would have a filtering capacity, right?
Yeah, so a lot of work's been done on introduced mussels and the negativity of, especially zebra mussels, the economics. They clog up power plant stations, so they need to. It cost millions to clean up. But ecosystem services are very similar to native mussels. In the Swan, I think probably I'd say the most abundant mussel would be the Asian bag mussel, which is obviously come over from Asia in ballast water. And it seems not to be too bothered living in sediment, which is, usually in our sediment we might have polychaetes and a few others, but not very diverse, not very abundant. Whereas these guys, if they set up shop, they're going to be creating all this habitat for other invertebrates.
Right, so they've sort of filled a niche that was not filled.
Yes.
Right.
Yeah. Okay. I'm not saying they're good. I'm just saying that they're doing good.
Interesting. And they have
other negative impacts apart from like economics?
So, for example, in the Peel, where I usually collect my Mediterranean mussels for my research, I found some of those spots are now overrun by the Asian bag mussel. So they do like hard structure as well.
Right, so they're out competing.
They're out competing the native species.
Interesting.
Yeah. And I guess, with a lot of introduced species, they don't have natural predators. We haven't really found any evidence that native fish or crabs will eat the Asian bag mussel. And maybe part of that, when it's on the sediment, it's called a bag mussel because the abyssal threads sort of cover the whole shell. So it's
they can't actually get it.
Yeah, so they might not even see its food. It just might look like a mat.
Right. Until they learn?
Well, we could try to teach them.
Yeah. Fish school. Right. Alan, this has been great. I've had a great time.
Same.
Yeah. Yeah. And I learnt so much today. I see you every morning.
Not if I see you first.
Well, thank you for very much coming on today.
Thanks for having me. Been a pleasure.
Paramount importance.